Okay, so without further ado, please join me in giving a warm welcome to Dr. Prashar.
Thank you very much. Right, I'm going to be very slow. Let me know if you think I'm too fast, okay? Please. So, what I'm going to do is, I'm actually going to go there, and I think I can still work fine. So, the idea behind my presentation today is to see how we can actually use different approaches using imaging sensors for building up some kind of a high-throughput resource, right? So we'll give you examples mainly in the field, and then I'll also give you examples in the vertical farm. Okay? Let's make a start. This is the group which I lead. So I lead a group called Agriculture and Animal Science. It's a group of nearly 82 people at the moment, and out of those 82, nearly 23, 24 are kind of PIs, like supervisors. Okay? And the main things which we cover in our group are linked to health and welfare, which is crop health and animal health and animal welfare. We look at quite a lot. This is my area which I cover a lot, which is data and circular digitalization. The idea behind there is to actually get the data and how we can actually move them around to actually improve that data analysis. And that's why it's called circular digitalization. Right? That's the idea. Then how we use all of these together into land use and food security. Okay? Can we actually build something towards land use and also food security? By using this, improving the land use and improving the food security, can we actually link that to how we can actually build up more like a nature-rich environment? So we actually benefit the environment instead of just looking for the benefits which we have. So it's the idea behind the whole thing. Okay? So just a couple of examples which I want to just share with you, which is some of the projects which we work on are linked to soil health and function. Pigeon I know now is Pigeon is my student, that's why I say Pigeon. Her name is Patacharya, right? So she's actually started working on soil health, that's why I thought I better mention it as well, right? So that's mainly some of my colleagues. I am only involved in one project in soil health, which is like we are building up a 5G network in the whole farm, so that we can take any data from the soil and can be easily converted into a cloud database, and then we can actually use as we need. So that's the only project which I'm working on because I'm more from a data side. Then a lot of my work actually comes into precision agriculture, so that's what I'm going to talk in more detail now. The other one is crop health. We are actually linking that precision agriculture towards the crop health, mainly diseases, mainly you can say any of the stress components which are actually coming into play. It can be nutrient composition, it can be developmental stress, it can be anything, right? And all of these together comes into whole agriculture and how it actually benefits the environment, along with how this whole environment benefits the animals as well. So how do we actually look at sustainable animal production? So mainly, grass, when they eat, how they benefit, right? Then lastly, all of this can't be done if we don't have a dissemination. So engagement is a pretty important thing. So as a whole group, we are a very close-knit community, right, with each other. And for that reason, all of these things are pretty important to us. Some of the things which we have built over the last couple of years, we have actually looked at the fundamental science and we have taken them into more, this will work, right? Okay. So, what we are doing is anything from a fundamental science perspective, which can be plants-insect interaction, which can be photosynthesis, any of the crop performance or phenotyping, any of these fundamental science, how are we taking that into more like on-farm innovations or diagnostics? So we have created tools for quite a lot of different things. So we have created a chip, which is more like you can look at cell wall components. So that chip is available. We have actually looked at the health diagnostics as a group. We as a group. Okay? We are actually building up remote sport technology with a company called OptiSense. Then we also have fungicide-resistant diagnostics, and that's what I'm going to talk today because that's part of our work. And then herbicide as well. So some of the things which we have actually. So one of the companies which we are working on for herbicide resistance. That's this. So what I'm trying to say here is, it's not just about fundamental science. it's about how we actually take that fundamental science forward so that farmers and the community can actually use it as the benefit, here. Otherwise, there's no point in doing the science if you can't actually use it. So that's what the idea is, right? As per for the presentation and my work, right, we cover quite a lot of different things, okay? For some of the graduate students who are here, right? So we are covering crop monitoring. Crop monitoring is mainly for anything from more like a field level to regional level, right? And that is to actually monitor whether there's any disease, any stress in the crop, or any kind of a variability in the whole system, where satellite comes into play on like the regional basis, right? Anything for crop phenotyping, we don't go into crop monitoring, okay? Phenotyping is more about looking at a small canopy, plant, tissue, or a cellular level, okay? What we are trying to do is we are trying to actually incorporate both of these together, because the applications of both these technologies are same. So you can actually look at monitoring from a growth perspective, from a water stress perspective, from a disease health perspective, from a quality perspective, okay? Similarly for the phenotyping as well. The only difference is mainly this is at a small scale and this is at a larger scale, okay? So once you have got that information at a small scale, you can incorporate that into breeding, and once you have bred that particular thing, you can actually take more for a monitoring level. And that's what we are trying to do, okay? And most of the tools, most of the technologies which we use for both of these techniques are the same. That's one of the benefits we have. We actually have a germplasm for potatoes, and that's why I'm trying to combine what's the phenotyping and monitoring together into one, and that's the idea, okay? Am I too fast, or am I fine? Right, perfect. So in order to take things forward, what we are doing is we are actually developing a field smart agriculture toolkit, right? And in that field smart toolkit, what we are doing is we are bringing the component of monitoring. So any disease which is coming into the field, we are actually looking at that, right? Now, that is actually good. We are also linking with the breeding part, which is the phenotyping. So where there is stress and what type of stress, okay, monitoring. Where there is stress, what type of stress, and can we distinguish that stress from phenotyping perspective? Not about just that. The idea is if we can actually know that stress before, so pre-detection. So not just detection, it's about pre-detection. Another component which we have actually included lately into this pre-detection is about discrimination. And I will give you an example of most of these anyway as we go along, okay? So for example, this is thermal. We are looking at more from a phenotyping. So these all are different varieties, we're looking at that. This is multispectral, using a drone data set. We're looking at where the disease is coming, so more like a field monitoring. This is more like classification from a developmental perspective, so more like a field phenotyping and monitoring together. How does a variety behave? So I'm going to give you an example of this as well. And then this is more about if you're detecting something in the field, can you actually detect it before, or can you actually detect at the beginning of infant? So all of these together, that's what we actually, well, that's what my work is actually mainly on. So by combining all of this, most of the platforms which are available in the world are actually on either this component or this component or this component or this component. The idea of what we are using is we are trying to combine all of these components and create layers so we can actually build as one platform. And that's what we are doing as a field smart agriculture toolkit. So the examples which I'm going to give you is the first one, which is more linked here, which is more on plant breeding. The second one, which is more about, again, a bit of a plant breeding, but again, whoever is interested in drone technology, so I'll give that an example. Third one, it's more about plant monitoring, so more from engineering. Fourth one, it's more about hyperspectral cameras, if somebody is interested in that. Last one is machine learning. So I'm trying to cover all those components in the field, and then after that, I will give you examples in the work as well. So that's what we do. And I can skip slides if needed. Okay? So let me start with the plant breeding perspective. Mainly, for plant breeding, you need two things. One is a genotype and one is a phenotype. Phenotype as to how anybody looks like. right? Genotype as to what is the genetic architecture behind that phenotype. Okay? And when we are doing anything on a plant, you need to combine or associate these two components together, right? 10 years ago, our field sizes were pretty small. Now our field sizes are pretty big. So we have quite a lot of variety. Okay? So that's the benefit we have. Now, also, 10, 20 years ago, you can say our genetic maps, so the information of my genetics or your genetics was pretty sparse. But now, we have quite dense information. The problem here is the phenotype. 10 years ago, we were saying 1 to 5 going into the field. Even today, most of our traits are 1 to 5. Okay? So the idea is how to actually link those traits in more like a high-throughput technologies. Okay? So some of those traits like like yield, shape, or maturity, we have quantitative. Quantitative, I mean, is like information from 1 to 100 in every, not a discrete form, in every form. Okay? So that's the benefit we have for some of the traits. Okay? But we don't know half of the traits which are indirectly related to these traits. Okay? And for that reason, I am pretty interested in something which is called physiological traits. And now, there are two reasons behind this. Physiological traits, because most of these physiological traits have really been used in crop breeding. One of the main reasons behind that is because we don't have a measurement, or we never used to anyway, we never used to have a measurement for the whole field or whole field scenario for these physiological traits. We only used to look at 5 or 10 plants. Right? Things have changed, and they're actually becoming much, much better. Okay? The second is, whenever you're trying to identify a physiological trait that is linked to better yield, we need to have an effective, like a screening method. Okay? So for that reason, for me, one of the traits which is pretty good for me is called stomata. Okay? Now, what happens is, stomata is an interface between environment and the plant. Right? It transpires, CO2 comes in. Depending on what is happening outside and what's happening in the plants, those stomata open or close. Okay? Now, when those stomatas open and close, right, they are actually a response to stress as well. If there's no water in the field, what would happen is, although the plant water status is the same, if there is a stress in the field due to abscisic acid signaling, those stomata will close. Okay? Now, when they close, what happens is, can we relate this to any of the imaging stuff? Yes, we can. We can link it to a thermal camera. Okay? So this picture here is actually showing you that particular scenario. So what's happening in this case is, these two plants, these are actually trees anyway, right? A picture taken from one of the journals anyway. So these two trees are actually stressed as opposed to the rest of the trees. Why? Probably some kind of a stress due to disease or due to water, right? Because if by chance this is an example of water, right? What would happen is, if there's a stress, the stomata is closing. And when they are closing, the temperature is actually increasing because it's not transpired. And you all are engineers and evaporation process cooling, right? So that's what's happening in this case, right? So any of the plants which are stressed will have a higher temperature as opposed to any other plants which are not stressed, okay? And that's the simple technology which we use for thermal imaging. Okay? The second thing, what you can actually look at is, you can actually look at how that stress perspective is actually increased to or linked to yield efficiency. Because if stomata are open fully, right, that means everything is going more efficiently. Hopefully, the photosynthesis happens more efficiently. And because of that reason, what will happen is, there is an effect of yield. So basically, you will have a high yield as well. Okay? But if the plant is stressed, stomata are closed, they will not be having an efficient photosynthesis. That's why you will be affected as well. Right? So the only thing to know is, if there is a stress, higher temperature. If there is no stress, lower temperature. And the same thing which we actually used to look at some of the radar fields. Now, nobody did this before us. That's the benefit we have. So what we actually did in this case is, we looked at 180 different varieties of the fields. Right? And this is the first paper you can see in 2014, which came for the field scenario of probability, where we looked at so many varieties. Okay? So we looked at all those varieties, and we looked at different statistical approaches in which we can actually show what is stressed and what is not stressed. Right? So those are those 180 varieties here, and these are the temperatures at different time points using thermal camera. So we took images like different times of the day. We took images at different times during the period of life cycle of the day as well. Okay. So that's what we did. The cut short story is, what we did is, we looked at whether we can normalize the data, so we normalize the data. Then we tried to look at which particular gene is actually controlling that. Okay? So I'm not going to go into detail, but if you want to know in more detail, then you can come to me anyway. Right? So we found two QTLs, one on chromosome 2 and one on chromosome 7. The one for chromosome 7 only comes later in the season. Remember my previous slide, where this is a response to stress, this is the response to yield, because it's at the same location as yield. Okay? So those two locations. Okay? Now, when we looked in more detail, we found stomachal development and degradation, that's one of the genes, and the other one was Cas14, which is more like abscisic acid gene. Right? So these are the two genes which we actually found. Now, some of you might know microarray. We actually did a microarray. So you can actually see how this picture looks in a kind of a controlled glass house. So these are the ones which are stressed. In the normal picture, they don't look like stressed. These are the ones which are controlled. Okay? Using a thermal camera, you can easily distinguish them. Right? We gave them some more stresses, so you can see what happens. Then we did a rewatering. Okay? Now, cutting a long story short again, because I don't have time for all of it, what we found is the same genes in a microarray were the ones which were either highly regulated or showing a higher expression, or they were the one which was actually showing a lower expression when there was a stress in the field. Okay? So what I'm trying to say here is, basically, we took images in the field, we normalized the data, and then we found how we can actually go from a field all the way to a gene. So we know which gene is actually controlling that. Right? So I don't think any of the study up till today has actually done so much detail on it. So that's one of the examples we just showed. Right? So that was the first one. The second link to that is, how are these high-throughput tools benefit for a quantitative trade? It's a very good example, and if it doesn't make sense, let me know, then I will not put it next time. Okay? So what we are doing in this case is, some of this work is actually linked to pigeon as well. But some of that is linked to my other PhD students. Okay? So basically, this one is a fixed string which we use. And again, we started this work in 2015-16. That's what we started this work, right? 16-17. That's when we started this work. And what we were trying to do at that case is, we were trying to create 3D models of different potato varieties, right? Okay? Nobody, you can see any paper, nobody has actually created 3D models of nearly 200 varieties of potatoes or any other. Again, to cut story short, what is happening in this particular case is, this is the whole pipe. Basically, you're creating a surface model, nothing else, right? You're creating those surface models. You're creating a surface model for the soil, you're creating a surface model for the top of the canopy, and in this case, you can actually create a surface model for every 5 centimeters. So you can know how your canopy is going upwards, how it is going sideways, or whichever way you go. Okay? So we use a triangulation approach, right? And this is kind of a triangulation approach where your vortexes are actually joining together, right? To actually give you the 3D models of those canopies. This is actually giving you that 3D models, how they're going in the field. Okay? So we were looking at 200 different varieties in this case. So one, we actually looked at the canopy cover, and second, we actually looked at the volume of each of those canopies as well. Okay? What's the benefit? The benefit of this is, you know how the plant is actually developing during its life cycle. That's one thing. Okay? So what I mean by that is it actually growing sideways? Is it going upwards? Is it growing more like even growth all the way? Is it actually growing with a higher starting or lowering point? You have different ways. And when the plants are actually senescing in time, or when they are actually lodging, right? Now you must be thinking, what's the idea behind doing this, right? Any of these developmental traits is actually linked to disease. And the example of that is, if something is lodging from the start, the chances of disease transfer are much more as opposed to anything which is going straight up. Okay? So that's the bad. And that's what we're trying to do in this particular case, right? Now, in order to show you that 3D models, so these are one of the varieties, Bonnie. So we know that's replicate 1, this is replicate 2, and we know from those replicates how it is actually growing all through the life cycle. So we only had 4 kind of data points at this point. But you can actually look at these, you can actually see how is the height increasing, how is the area increasing. And based on those height and area, you can actually look at the maturity, you can look at senescence, you can look at how it is actually growing, whether it's lodging, whether it's not lodging, right? So we have this for nearly 275 models, am I right, Vijay? Yeah, 275 different varieties of potatoes. Right? So, you know, and these graphs are kind of replication of this, so you can actually see how the canopy is growing for all of these scenarios. Okay? So that's one of the examples where Bonnie, right, where everything collapses after the third flight, so there's nothing here coming in this case, right? Or there's another one called Bounty. You can actually see that it actually matures after this particular time. So you know that maturity actually happens after this point. So different colors here actually link to the height as to how high the flowers are. Okay? So anything you can say more into the pinkish range is quite high. Okay? And then we also have another variety called Nadine, in which actually is a, you can say, early maturing variety. Okay? So it matures quite quickly. Okay? That's what is happening. Right? So that's the example. Now, the benefit. What most of us have been doing up till now, they have been taking data in the field using a ruler or a scale. Right? When you go into the field, you take a ruler, you have 1 point. Right? Now, this data here is actually drone data. And this is a drone data based on probably 500 to 1,000 cloud points on the canopy. Right? And if you look at the maximum height, and if you look at the average height, you look at the difference of the range. So your average height for those varieties, 250 odd varieties, is between 0.2 to 0.3, 0.4, like 3.5, right? Roughly that for average height. And if you look at the maximum height, as to the toppest point, you can actually see the range is from roughly 0.5, 0.6, roughly in that range, right? So you can see the difference already when you're taking an average. So if you're going to the field, taking 1 point, right? If cloud data based on 1,000 points is giving you such a variability, you can see how much variability you are causing by taking 1 ruler point, right? So I'm going to show you this in the next slide, why I'm saying this, okay? But basically, anything we were doing based on what is happening in the field from 1 point to another point, we had adjusted our scale of really 0.5. That's nearly half, right? That says, well, our data is not very good, right? But it's not that data is not very good. It's not very good for the point that we are taking one single value using the ruler. So we don't know which data is good, which data is bad, right? What we found is, most of the research which has been happening over the last, you can say, 20 odd years, when we are taking those point measurements, is probably not giving us defined results, right? So I don't know how many of you know association mapping. You don't have to go into detail. Basically, the relationship between the genotype and phenotype, okay? So what we found in that case is, if you look at this data, and if you look at this data, right, you're having different regions on a chromosome which are actually controlling a trait, whereas those regions don't actually come here. So even that data is giving you quite a lot of variability, and if you're only having one measurement ruler, that's quite different to what you're getting. So all the genes which we have up till now might not stand in 5 years' time. Okay? So that's the quantitative approach which we have. Okay? I've still got another 20 minutes, yeah? Right, perfect. So that's the second example, why quantitative data is very important, right? Coming back, the other benefit which we have from this, as I said, we can look at senescence, we can look at lodging. I'm not going to go into detail, but basically, just going to tell you, we've already created a model where we actually know which one is actually maturing early, which one is actually senescing early, or which one is actually both senescing and lodging early. So, there are different examples. So we have actually created like a cluster-based models and also random RF models, random frequency models, right? Or random forest models, sorry, not frequency models. Random forest models are looking at those as well. Okay? So we can differentiate which particular variety is growing senescing early, which particular variety is actually lodging early, which particular variety is actually senescing and lodging together or early. Okay, so both of these together as well. So different things you can actually do on this case. Okay, I'm going to cut the story short here. I'm going to go to the third one. Okay? Any data from the imaging sensors. This is kind of a repetition of what I'm actually showing here, but I will show you in a bit more video. Okay, so what's happening in this case is we are looking at stress. Okay? As I said, anything developmental can be linked to disease as well, right? So this is kind of a multispectral data. This is actually pictures right? So multispectral data, which is taken again on the field. What we are trying to do here is we are trying to create a model for blood count, and we're also trying to create models for looking at different indices, whether those indices can be related to any stress. And that's what we're trying to do this instead. So this is more like a pre-processing and post-processing pipeline, which we're using for that particular case. So this example here is mainly, this is your input image. You create a soil mask, and anything which is soil is actually excluded. Then you have your plants, and then you're trying to create a plot shape to actually see which particular plot you're looking at, and then you actually create those kind of 3D structures or surface models for those structures. Okay, so you create them. Once you have done those, what we're trying to understand now is what is the number of plants which are actually coming, and at what state we should be. Right? So we actually created those models. We looked at those indices in different scenarios, because those indices actually telling us where in the field there is a stress, NDVI, NDRE, GNDVI, any of those, right? Those are any variability in those. So this is actually a plant count. So this is actually counting the number of plants which are actually in those groups. So 3 in here, 3 in here, and so on. So you can actually count the plants, but it's depending on the stage as well. Once the plants overlap, it's slightly harder to do it, but those models still work. Okay? And these are the indices which we actually created based on that. Oh, sorry, I never knew this was a touch screen as well. Okay? Is it? No, maybe it just went on. Right. Anyway, so what we can actually do in this case is what we looked at. Those are the RGB bands and also red edge and NIR bands. Using those bands, we looked at the vegetative indices, different vegetative indices, and then we looked at what those vegetative indices are actually giving us, or what are the variability of different, you can say, plant varieties for those vegetative indices, and are they actually related to any type of crop development or any kind of stresses. In simple scenario, that's what we are looking at. Okay? So we're still doing it, but we have those variabilities. Okay? And I'm not going to go into detail again. But basically, you can look at those varieties which we were talking about and look how that variability is actually happening all through. Yeah? And one of the most major ones which we are actually interested in is RVI. The NDVI is good depending on the stage where you're getting it. At certain stage, NDVI doesn't work, then your NDR is actually giving you a better value. And that happens when you have a lot of data. Right? That's the idea behind it. Similarly, your NDVI is actually related to some kind of a photosynthesis structure as well. So your PSRI is behaving the same way to a certain extent for your NDVI. So it's just the variability for monitoring. We are looking at those cases. Now, this is for the varieties, but we can do the same thing for actual field scenario as well. So both for phenotyping and also for monitoring at the beginning. Right? So this is one of those examples. Other thing, which I'm not going to detail, using those, what we have actually done is, I will show you in a minute anyway, but using that, multispectral camera, what we have also done is, we have actually looked at, can we actually use a machine learning approach to distinguish those varieties, okay? So we have managed to actually do that by using that multispectral camera to an accuracy of nearly 68 different varieties for 97.1%. Now, not all the farmers actually have multispectral cameras. They can't use it. Okay? So I'm taking you to the next one, which is more like, can imaging actually help us? Well, kind of classification to a certain extent, but I will take you to the next example in here, right, later on, where we're actually looking at those classification models using RGB, because farmers can actually use that. Okay. Once we have that stress incidence, right, we also want to know whether it can actually help us with PE detection or not at all, right? So for that reason, your multispectral doesn't really work, because the number of bands are very limited. You can't distinguish between different diseases. You can't distinguish between different parameters of the same stress. Okay? So for that reason, we're actually looking at hyperspectral. I'm not going to give you a disease example here, because that disease example is still happening. I'm giving you an example here. So that disease example is also slightly more complicated, because we're using attention-based protocol for machine learning, and this is slightly new protocol, so that's why I thought I better not go into it. I'm going into more journal here. So these are like the hyperspectral images, okay? And this is only visual near infrared. We also have shortwave infrared as well. I'm only looking at visual near infrared. The benefit of visual near infrared here is because, simple terms, what we are looking at is we are looking at greening. There is a benefit of using shortwave infrared, but not to the same level as VNI are in this case, because what we are interested in is whether we can actually detect greening before. And you're using shortwave infrared if the chemical composition is actually very good. Then we will have a benefit of shortwave infrared in this. So this is only restricted to VNIR. Okay? So this is kind of a raw image, right? And we crop those images, doesn't matter, we mask those images, so we have those different potatoes. Okay? Now these potatoes have greening, these potatoes don't have greening, these potatoes also have disease to a certain extent. So we have to discriminate those things as we go along. Okay? So there are different ways we actually try to do it, right? And those different ways are, we try to normalize the data in the first place. We use specific calling as first of the polynomial information, right? So that's what we looked at, okay? Then we normalize using SNB as well, okay? And then we looked at first order derivative. So these are like a pre-processing steps which we use to look at whether we can look at the variability and whether that variability is giving us something or not, okay? But in certain cases, you can actually look at your light green and early green or your full green. You know, certain spectra are actually looking at those and you can actually distinguish those. So 694 nanometer, that's actually distinguish those, and then 688 is actually distinguishing between light green and early green. So we get a bit of information on that. A step further, so more like a pre-processing or normalization. Step further, what we try to do is, can we actually look at that detection just using normal k-means clustering and also PCA, right? Principal component analysis. Principal component analysis didn't really work. You couldn't actually kind of detect each and everything, okay? Your component 1 was giving us mainly the light. Your component 2 detected sun greening. Component 3 was a mixture of different things. And component 4 will give us a light green. Yeah, sorry, 5 I think it was here, right? That gave us light green. So you couldn't really, so normal computing approaches maybe didn't do it, okay? And we didn't go into any of the deep learning approaches in this case because we didn't want to complicate things, because this is a simpler model, right? So we just wanted to approach it from a machine learning perspective and also want to keep it simple in order for us to learn as well, okay? So when we did the 6-mean clustering, or such, k-means clustering, you can actually see, we can look at different diseases, types. We can look at light green, green, and kind of a disease too. But still, there are some mixed information where we can't distinguish as well, right? So instead of going for those approaches where we actually just tell, well, we just say to the computer, you actually distinguish it, we try to use a supervised classification, right? So this is a supervised classification. What we did in this case is, we actually told, this is the region which is related to this disease, this is the area which is related to greening, this is the area which is light green, and this is the area which is non-green. So those, we actually told the computer, this is what we do, and you should actually create a model based on that. Simple, right? And based on that, you can actually see there are different spectra, so we use the both raw data and the SNB data, we actually look at those, okay? Now, cutting the long story short, right? And you can actually see in certain scenario that you are actually able to distinguish based on those areas between early, light green, controlled, green, and disease. So those are the 5 categories we are really interested in, right? So we actually created a scenario where we actually asked the computer, these are the problem areas. Okay? So then, using that approach, what we did is, we actually created a data. So that's like a supervised learning, which we did up to now. But then we said whether we can apply that supervised learning on some kind of a temporal grading. So what we did in this case is, we took different potatoes, right? And based on those different potatoes, we actually left it to green for certain scenarios, we left it to disease for certain scenarios. So day 7, day 14, we saw anything which is slightly greener on day 1, does it actually increase in greening, or certain parts actually increase in light green or dark green. Okay? So we're trying to distinguish between all of those classes. Right? So this one here actually tells you those supervised learning based on that temporal. And once we have actually done that, what we found is we can easily, using those SNB images and raw images really didn't work for the training set, but your SNB based images actually work for those training sets, right? And those actually gave us classified images when we actually used 2 approaches. One, SVM, right? And second, spectral angle mapper, right? So both of those machine learning based approaches actually easily distinguished with the areas which was like light green, which was diseased, which was slightly more green, and which was fully green. So we actually create that classified images based on all the supervised classification which we've done using the temporal and also the spot. So it's distinguish those things. So that's kind of anything which is turning green can we actually detect before it is light green or even before it is actually turning green. So that was the idea behind this. And we can actually use it to some extent. So that was the simplest example in hyperspectral I would take because there's a lot of bands and there's a lot of processing happening in this space. Okay? Let's come to the last one where I'm going to look at the stress incidence and classification model. You can stop me any time you want and say, you're finished your time. Right? So classification models, what we are looking in this case is where we are looking at, again, machine learning, fully machine learning in this case. We are looking at daily distinguishing those 250 varieties. Right? And the idea behind this distinguish is because if we can distinguish those 250 varieties, then we can actually go into potato field or any of the farmer's field and tell them, okay, this is a variety you're growing, this is how it is actually developing, and these are the diseases, stages, when we will have that disease. That's the idea, okay? So what we have done in this case is, so if anybody is familiar with machine learning, we're using different models, Resident 18, Resident 50. We also use VGG 16. We actually use 16 different models, but I'm only showcasing these 16 different models we looked at. Okay? So these are the backbones which we use in that classification models. So when we looked at those models, so these are nearly your predicted labels and these are your true labels. Okay? Using that confusion matrix, what we found is using those models, right? So this is Resident 50, actually, which is the one which actually worked the best. Okay? In our case. VGG 16 didn't work perfectly. Well, that it worked the best, Resident 18 didn't work that much, but still fine. So what we found is that most of those 25 varieties, so we started with 5 varieties, and we only gone up to 25 varieties. Those 25 different varieties are actually easily detectable using these models. So we can actually distinguish those 25 varieties. Okay? And it's not just about distinguishing those models, it's also about how much computing power you need for these things as well, right? So 25 varieties is good, we're going for 64 now, and then once we have done the 64, we're going for 128, and then we'll be going forward to the 56. So we are increasing, but the computing power actually increases quite a lot. So we were also looking, I didn't show you that slide anyway, but that's fine. We were also looking at how much computing power is actually needed. And that's just based on RGB images, nothing else, just RGB. Right? Right. Now, taking that slightly further, we are actually also looking how those things, another example on machine learning, can we actually distinguish different types of weeds as well. It's not about just detection, it's about discrimination of different weeds as well. Okay? Cutting long story short, because I think I'm running out of time, alright? I'm okay? Or 10 minutes now? Right? Does it make sense, anybody? Am I too fast, too slow? Yeah? Perfect. Okay. So just to cutting a long story short, okay? What we were trying to do, we were trying to do two different things in these weed models. Okay? So these weeds are actually growing in potatoes. And most of these weeds actually grow at the same time. And there are different species of weeds. We were looking at six different species of weed. The idea was, can we actually discriminate them? That's one thing. The other thing was, if we were growing them in a glass house and in a field, does the models which we create in the glass house, are they applicable to the field or not? Because nobody has looked at those things, okay? I think you know my answer, right? But this is normal methodological approach which we actually were using. So you actually have your data collection, you actually have your pre-processing, and we actually approach another thing in the middle, which is more like undersampling. I will come to that in a minute anyway. Then we did the feature selection and then classification and analysis. Again, this is machine learning, not deep learning. We have done deep learning on this as well, and deep learning is much better using CNN, mass RCNN approach. The only reason I can't show you that is because we put in a patent for that one, that's why, right? But I'm happy to discuss that, right? So this is only machine learning, right? So what's happening in this case is, so this is kind of a machine learning approach. We have the data collection, processing, and the sampling, and we looked at two different ways of classification. One is KNN approach, and the other one was cluster models. We actually look at the timing of those, right? So, and we looked at random forest as well. So two different approaches, KNN and random forest classification. Okay? The idea was not to just look at whether we can discriminate and classify. Also, what is the best time to actually classify those weeds? If farmers are spraying it, what is the best time to spray those, right? And this is just a data collection approach. This is undersampling. So I don't know whether all of you know undersampling, what it actually means. Basically, instead of using a manual approach of actually creating, so when you're actually doing, so basically two different approaches together. Okay? The first one was BG-based approach, where we actually tried to look at a certain weed, estimate those weeds. And then we actually use undersampling approach, where we actually try to undersample this so that it actually creates a boundary and discriminate that weed, right? Just a way of annotation. That's what we did with BG to annotate. Okay. Once we have done that, right, what we found in this particular case is two different things, right? When you're doing something in a real scenario, in the field scenario, okay, and when you're creating any of these machine learning models, it works best when you're having cars, you're having buses, you're having cats, you're having dogs, right? Because your number of samples are always kind of similar, right? When you're doing something in agriculture, things are more complicated, much more complicated. Environment, one of the factors. Other major factor is, if you're taking, for example, weed data set, right? If you look at the figure here, right, on the top, right? You can actually look at the percentage of those weeds, which are actually 0, 1, 2, 3, 3 of those weeds, okay? Some of them are actually more, actually, sorry, that's what I was wondering, what these actually have. I think I missed a slide here. Yeah, when you're doing anything in the field, sorry, I didn't miss that slide. So basically, 1, 2, 3, 4, 5, 6. So the amount of weeds actually vary. Your background is 75%, but amount of those weeds actually vary. And when that amount of weeds vary, your classification models or discrimination models don't work as perfectly as you think. Okay? So that's why what we try to do is we try to actually use the pre-processing approach to actually create a scenario where we have similar kind of a data set. When your data is similar, then your classification models actually work perfect. Okay? And that's what I'm saying. Things are easy for cats, dogs, buses, cars, because you can actually bring anything from outside. But when you're doing something in agriculture, it's slightly different. Okay? So basically, that's what we use. When we do that, what we found is, so we use a 10-band camera, in this case, band 0 to band 9. What we found is, if you actually look at it, right, so two different scenarios. Okay? One, under the same data collection environment, we still have different feature selection methods. We actually selected different feature bank. So everything was not the way we were, right? So that was actually happening under the same data collection environment. Even in the glass house, you can actually see, so your greenhouse here, your orange colors, you can actually see different bands are giving you different information, right? Then, when we actually want to transfer something from the greenhouse onto the field, the same feature selector, but different data selection environment generated really results as well. So, basically, you can't really apply what's actually happening in the greenhouse to what's happening in the field. It's easy to say things are working perfectly when you're generating something in the glass house and you're bringing it forward to the field, but no, it doesn't happen. There are some features which you can actually still use. So, for example, these features you can actually still use. That feature there you can still use perform. And those features can still work, but some of the other features are actually quite different. Okay? So, well, looking at those things, your KNN still give you quite good accuracies. It gave you quite good information based on different. So we use RFE, we use RFPS. You don't need to go into detail. Don't worry, okay? I'm not going to go into detail. Don't worry. But basically, what I'm trying to say here is your KNN or nearest neighbor means their KNN approaches were actually working the best approach for your classification model, right? Either it was for your glass house or your field. But your field scenario was much better as opposed to your glass house. Benefit here was because we had that variability in the field as opposed to your glass house. That's why it actually worked, right? But the rest of the things are mainly about SPMA is fine, random forest is fine, but KNN was one which was working. Although their accuracies, their different information was good as well, right? The other thing was, when we actually trying to delete a certain number of bands using all these approaches, the 10 bands were actually working the perfect or giving us more information. Naturally, that's the case. The more information you have, more you will be able to do. Okay? So that's what it was actually. This is your 10 band. Okay? So yeah, I'm going to just classify that based on the machine learning model because there's a lot of detail behind it, but I just portrayed that as one classification. Now, the same approaches, I'm only going to take another 2 minutes, and then I'm going to stop. The same approaches, what we're using is, we're also trying to apply in the case of vertical farming. So this is a vertical farming. Okay? So what we're trying to do is, in this particular case, so we already published this one, that's why I thought I would put it in. Right? Infrared thermography for plant stress detection in the vertical farming. Okay? It's not a septic in this one yet. It was it's getting accepted in another one now, but I put it there because that's what it's right? So what we're trying to do in this case is, we are looking at, when we are growing something in the vertical farming, right? So you have different traits in a vertical farming. We were looking at the variability in the trait, and we are also looking at how, if you are applying some kind of a water stress, how many hours after that stress is there, we need to apply water as well. Okay? So there are different scenarios we're looking at. So irrigation scheduling, which we were talking about. And also, we are looking at what is actually happening from a disease perspective as well. So there are different models which we have actually created, right? So I can talk about, I think I'm going to see you anyway, I'm going to talk about these in more detail later because I don't think it will be applicable to everyone. But we are using the same approaches in the vertical farming as well. So not just that, we are creating those 3D models and we are looking at those colorimetric attributes, so different indices in the class, in the vertical farming as well. Okay. Thermal and phenosphecs, using those 2 approaches, we are actually creating those colorimetric attributes and also the structures of the plants as well. Right? And lastly, we're also looking at how UVB affects the plant in the vertical farming system as well. So how do we actually look at photosynthesis, how we look at different hormones and stuff like that. And those are actually linked to last one, right? Sorry, I'm squaring the 2 now. Go on. Maybe that's the next one. We are also looking at, with regards to, so we're using quite a lot of different, we can say, plants in this case. We're trying to look at coriander, we're trying to look at dill, we're looking at curry basil, and we're looking at cumin. Okay. So we're looking at carotenoids, we are looking at anthocyanins, we are looking at chlorophyll content. So we're looking at, we can say, mainly any of the metabolic traits, and any of these colorimetric traits that we can actually distinguish those using machine learning. So we have used different approaches to actually do an assessment of these antioxidant components for those things as well. So that's another one for what we found. Okay. Next one, please. Sorry. stop working, right? Lastly, just last 2 slides. This is one of the newest projects which we actually just got, well, kind of 6-7 months ago. There are 2 different things which we are trying to do, right? Now, when we do something in the vertical farming system, what people are morely interested in is actually just yield. We're not interested in yield. Yield is one thing. What we are interested in is more like a nutritional quality of those yields, right? What I mean by that is, we want to harvest the plants when they have the maximum nutritional quality instead of the yield. So this particular funding is part of an innovation hub which we built in Newcastle, which is called Vertitech Newcastle. And this is one of my degrees, right? Because I'm interested in Vertitech quite a lot. So what we're doing in this particular case is we are looking at how pathogens are affected. We are looking at how we can actually approach using AI models or imaging data sets. We are also looking at how we can actually look at different kind of vitamins and or any of the metabolites, and can we link that to the to the imaging resources. And lastly, we are looking at how different blood holders are actually affected this as well. So basically, what we're looking at the nutritional quality along with the yield component, and can we actually use imaging instead of using chemical analysis. Because if we do that, that will actually benefit us instead of taking the destructive sampling from any of these plants and taking it to the lab, if we can do something with it. So that's what we're doing. And I can't showcase everything in this one because it's a big project and there are a lot of people working on this, right? One of the components which we are very interested in is reduced potassium, and that reduced potassium is because anybody who has a kidney problem, they need a reduced potassium. But at the same time, they need all the other nutrients to the same level. So we're trying to do that based on this particular project, right? So using many different approaches, meta-analysis, for example, ICP-OS, and any of the HPLC analysis. So we're linking the data sets, so we can actually link that with the machine learning approaches. That's what we're doing. And using RGB spectral data sets or hyperspectral data sets. That's what we're doing, right? Next one. Yeah, I'm going to stop here. Sorry, there was a lot of information in all the slides, but I'm happy to share this. But it's not just me. Over the years, I've got quite a lot of PhD students. I mean, this is kind of a list, and the postdocs here who have been working with me. And one of them is still not a doctor. I want her to be a doctor soon, but she is here. Hopefully she will get that doctorate degree soon, right? And then I collaborate quite a lot with University of Birmingham. I collaborate with Surface Solutions, one of the companies. Phyto is one of the companies I've been collaborating for the last 2 projects and been of immense help. And then GFM Institute, I work very closely with the University of Kentucky and Durham University. Again, quite a lot of funding coming from different sources. Otherwise, it's not possible to achieve it. And at the moment, I've got 16 PhD students, and I have no idea how to fit them into all of these studies. So that's why I'm here. Thank you.
Thank you for Dr. Prashar presentation. It's very, very nice presentation. Your work is covered with many topics in agriculture, including engineering. So at this time, we would like to ask anybody who wants to have some question, please, with question time.
Can I quickly grab a water? That's fine. Yeah, go on. There.
It's actually a lot of information for me. It's very nice to see you, Professor. So I'm Luke from Agriculture Engineering Department. So I really want to know is when you are doing particle farming and you are detecting with the camera. So I really want to know is there how can I say a specific angle to detect the plant? So maybe if very large scale, we cannot. There will be a blind spot for the camera. So is there any?
Yeah, I think that's a very good question actually, Luke. Right, thanks for asking that actually. So there are 2 different things which we are doing, right? In one case, what we are doing is we are taking the trays out and bringing it to the camera. Okay? And that is just what we done, right? We're not looking at stereo vision here, because when you're looking at stereo vision in this particular case, what's happening is because there's an overlap between, it's hard to actually distinguish it, right? So one of the newest models that we are doing at the moment using hyperspectral is we are trying to actually create classification masks for each and every plant in that tray, even if they overlap, using masking approaches, right? If that's successful, then this would work. Now, the PhenoSpecs is not working this way. The PhenoSpecs model works this way. And that's one of the problems which we have with the PhenoSpecs. When we have the overlaps, it's not taking half of the images. And that's why the vertical is actually perfect. Now, that would not happen in a stereo vision approach, because in PhenoSpecs you only have one camera, and that's LiDAR based and multispectral. Okay? If you're using a stereo vision approach, you're taking each and every angle. The problem then happens is actually amalgamating that data and increasing the power of the GPU. Right? Otherwise, if we can make things simpler, how do we have this?
Yes, sir. Thank you for the presentation. I want to ask you about the monitoring and machine learning. In case that we found some similar phenotype of the symptoms. For example, if we study about the south stress and the plant appear the yellow leaf, and also it probably affected by the nutrient deficiency. So how we detect this kind of phenomena?
That's a good question actually. So that's all about interaction, right? So salt stress is one of the components which I haven't looked at in my life. But that's where I think I would say hyperspectral would work. But the problem in that case is when you have any of the salt components, the signaling approaches, even your potassium channels, are too close. So your nutrient composition would not change at all unless you dilute that salt. So all these factors actually work when salinity is not a problem. So I think first thing which you might have to do, you can distinguish between salinity and nutrient deficiency, that's not a problem. But the best approach in that case is to actually look at the EC of the water and actually decrease that first instead of going for the plant nutrient deficiency. Because if there is a salinity, for example in the fields, do we have a lot of salinity in Thailand? A lot. Yeah. Oh, yeah, in your campus as well. Okay. The problem in salinity cases, if you have a saline soil, you also have a drought. That's the problem, right? And when there is a drought, as I said, those channels do not take water, right? So you can see the stress. To discriminate that stress is going to be very hard. I'm not denying it is going to be hard. We can actually try hyperspectral. I've never tried it because we don't have that much salinity. Well, it's the same when you have a lot of cation exchange capacity. If you have a lot of nutrients, which is over-nutrient, you will have a saline kind of scenario as well, right, to some extent. So it would be kind of, yeah. I don't think I can fully answer the question, I agree with that. But on the other hand, I think we should be able to look at the spectral signatures which might distinguish saline and nutrients if we do a controlled experiment and then see the result. That's the only way I can see that. But saline is a problem. Yes.
And as I am a physiologist, sometimes we found some difference of plant response. For example, in the heat stress, we found that mostly the plant should reduce the stomatal to respond to the heat stress, but our case we found that the plant try to enlarge the stomatal to facilitate more air inside to reduce the temperature. So our plant is Kamphaeng. It's orchid. Sorry.
Oh, it's orchid.
It's a kind of Kamphaeng, and the stomatal is open at the night time. So maybe you can note in other case, if the plant that try to activate and adapt themselves to the heat stress by increase the stomatal, so it is effect to the
Yes.
yeah.
That's
idea.
again a good question. So this is something which we found as well. So We haven't found this in potatoes, but I know working with somebody else on orchids, so somebody in Vietnam, we had a good relationship with them. So in orchids, what's actually happening is, it's not just the stomata, it's the extra roots, which come out as well because of the heat stress. And same thing happens in potatoes as well, actually. So when potatoes are actually stressed, they have a secondary shedding as well. Now, the number of stomata actually increases. I don't think the size of stomata actually increases in this case, in the orchids. It would be nice to look at that one, because that's what we found in orchids in that case. But the number of stomata will not hinder thermal imaging, because what you're still getting is you're having a microclimate which is quite different, and your variability of a control and your other plant is going to be quite different. So that would actually work perfectly for even for the thermal. Yeah. Does it make sense? Yeah.
I don't think that's working.
Yeah, this also works.
Use the one at the front.
Thank you for the presentation. I'm a PhD student from Rice Science Center, and actually I'm working with this bacterial disease in rice. I would like to know if the imaging can be trained to detect different kind of diseases. And another question is, if the plants in the field have the different kinds of stress, for example, drought, or maybe another disease or insect stress, how the imaging can detect?
Good question. I haven't actually shown you that. Well, I used to work on this. That's the thing which I'm doing. So I used to work in collaboration with Gilly, but we just looked at the drought of that rice. That's what we have. We have one papers with that Gilly. So what we are doing in potatoes now is we are looking at light, PCM, viruses, drought, and nutrient composition, all in one go. Right? We're doing it in a controlled classes at the moment. And then once we found the spectral signatures which are actually available for these things, we take that aside. Same approach will actually be applicable in the works. Right? Now, there are two things to approach here. Whether you really want to discriminate the diseases or not, depending on the type of disease. Right? Mildew, or any of the, let's say, bacterial disease. You're looking at bacteria. In bacterial case, your stomata will be affected quite quickly. Right? Anything in bacteria will be affected quite quickly. And when you're trying to distinguish that from the spectral signatures, it will be quite critical. So we did a very good experiment, and that was on PCM, potato cyst nematode, which is not bacterial disease, but it's a cyst nematode. But it's actually linked to drought. What I mean by that is that if you have a drought, you really want to distinguish it from PCM, right? Bacteria is slightly different because you will actually directly affect your stomata. That's a problem here. But your spectrum, so your thermal will not give you any variability, but your spectral signatures will actually be variable, especially the hyperspectral. And we're doing this in potato at the moment, and if it works, I will let you know. So we're looking at 5 different diseases and 2 different stress scenarios. But in hyperspectral it would. The only problem is you need a lot of variability in order for machine learning to work. That's the thing. You need a lot of data as well in this case, because you. On the other hand, why to complicate things? Right? For 2 diseases, drought and your bacterial variant, I think it should be fine for you. As long as you don't have any other disease interacting with it.
What if we have combined another nutrient deficiency?
Nutrient deficiency will behave in a completely different way. I'll tell you this now, right? Because the spectra of your nutrient deficiency in the shortwave infrared is actually quite different for your nutrient composition as opposed to your video infrared. Yes. So your SWR will give you much more information on your nutrition deficiency. Your VNIR will give you more like the visual characteristics, but your SWR, your cellular structures are changing. So you tap into deficiency question. whereas your bacteria, based on the drought, that would actually have both as well. But the drought would just be in the first instance.
I have small question. Now for your the motoring system, and you know you work with the potato. Do you monitoring on the yield on the yield prediction?
So yes, so we are using a model called ESAT model at the moment. So ESAT model, I think, quite widely used. So there are 3 different models which we are trying to apply. One is the accent model, which is the Australian model. The second one is a Koch model, which is a Cambridge University model. But that model is very simple. It's mainly on the radiation and mainly on let's say radiation as an amount of light, right? So that Koch model is normal. So what we're trying to do now, and nobody has done this before, one of my PhD students, Sheffield, right? What we're trying to do is, in a DSAT model, up till now, it's mainly the environmental parameters which have gone into play for yield prediction. What we're trying to do is, we're trying to incorporate the UAV-based data set into the DSAT model. So we can actually look at how the variability in the 3D structures is actually affecting the yield. Because that gives us quite a lot more information instead of just environment. So yes, we are doing it. So DSAT is what we are doing.
We have time for one more question. Thank you.
So, my name is Joe and I'm from Kamphaeng University, and I have a question. I've done my senior project in internal disease in mangoes. So I just took a picture and analyzed the photos. But I want to currently work in my master's. I want to do like hyperspectral image to detect the disease severity in mangoes. So the question is like, in your opinion, like which type of imaging will be better, like to detect the disease, like thermal or hyperspectral or something?
So I think, how long is the MSc project? How long? Is it 6 months, 3 months?
For me, I'm just a 4th-year student, so maybe next year I'll start.
So in 1 year, yeah. So I mean, if you want to just detect and diagnose, right? I would say you can actually even detect it using thermal, right? But if you want to actually look at pre-detection, your multispectral will not work. I can tell you this now. Because if you really want to look at the multispectral, you need to look at specific bands for that anthracnose in NASA first. You know, there's a NASA web page for quite a lot of different indices, right? And somewhere or the other, if you can actually find that index, which you can create on your own by looking at different bands, or if you find something who has done before, which is linked to anthracnose in any other crop, then your multispectral will work. Because then you know which particular bands you have to look at. Just using a 5-band camera in a multispectral, it's pretty hard. That's why I built that 10-band camera, which we are using now, right? Because in those, the problem with the 5-band camera is your wave bands are quite wide, right? And they don't give you discrimination information. If you're using a hyperspectral, that's the benefit, because you have those narrow bands, right? That gives you that discrimination. Unless you build your own, like a 10-band spectrum bands or something like that, then you look at those. So if you just want to do the detection, I would say just go for thermal. If you want to look at pre-detection, probably a day or so before, I would still say go for thermal. That would actually be easier for you, right? Incredibly. But if you really want to look at and you're discriminating that from something else, then I would go for hyperspectral. Because hyperspectral is a good tool, I'm not denying it. It takes a lot of energy, as in human energy, in processing and producing and everything, right? And it's good to learn new tools, but is it really applicable to the farmers? It is only if you have a certain spectral bands which you can discriminate and then create a multispectral for that particular disease. Then it's actually very good. If you can't go to that level, and if it is just about, whether you cover disease or no disease, just use it. And it would work in the same way for any of the diseases, because what you want to know is whether the plant is stressed or not stressed. But if you want to know if you want to understand the spectral signatures linked to enzymes, then you will have to. There's no other choice. Right? But on the other hand, you don't have to go for the short wave in this case. You can actually just go for the short wave. Because in the diagnosis as well, you will have probably your chemical properties will change later on, but in the first instance, it's the cellular structures will change.
Ankush, thank you. We have one last question.
Okay.
Thank you for your fantastic words. And I know that it's tons of tears, effort, and words. So I just actually wanted to mention a little bit about drought. Actually, the plant and different varieties, they have many strategies like drought tolerance, drought resistance, drought avoidance, and drought escape, right? So I think you maybe have to observe every stages of the rise of the plant or crops. And my question is, if I have to establish the lab and I have a low budget, what kind of technologies or portable equipments that you will suggest? Yeah, thank you.
Yes, good question. If you're just interested in drought, I would use a soil sensing approach. And that's the cheapest option. There's no point in doing it. So you can actually create, so we are doing this and you can do this. We're actually creating a machine learning approach for soil-based sensing. So what we are doing in this case is, I didn't show this in the slides. What we're doing in this case is, we are using one of the soil sensing probes, right? And those probes give you EC, as in electrical conductivity. That electrical conductivity is linked to many different things. The kind of excited capacity, amount of moisture, salinity, but you can't use soil probes and create a model if there's a salinity. 100%, right? It's pretty hard. Because your electrical conductivity is going to be always high if you have a salinity. Okay? So what we're doing in this case is, we actually use that electrical conductivity and your soil temperature and pH to link with different nutrient-based compounds. Okay? So instead of using the RGB data set, instead of using any of the data set, those are numbers. We don't need that much GPU, and those numbers can be linked to different nutrient conditions. The only thing you will have to do in this case is, you will have to link those with some of the techniques which I mentioned. That is ICP-OES, ICP-MS, and look at the implementation. More variability you have, more data set you have, you will actually create that machine learning. And that will be the cheapest option now. The sensor will cost you probably around $250. And instead of actually using a cloud-based sensor, you will go to different places and measure the data, take the soil sample, do the chemical analysis at the same time, and see how it is actually going to impact the soil. And then you can change the development. That would be the best.
Okay, thank you for all questions. Thank you to Professor Prashar.
I say Ankush. It's actually easier for me.
So, design design. So, right now, on behalf of the Faculty of Engineering and all participants, we would like to thank you to the other for our presentation, great presentation today.
Thank you.
Thank you so much.
No, thanks for having me. I appreciate it. And hopefully it made sense. Right? I knew there was a lot of slides. I did that. But yeah, I tried to skip for a while. So hopefully it makes sense. Thank you. Thanks for having me.
Thank you so much. And at the final time, we would like to take a group photo with everyone at the Faculty of Engineering. Please go up over there.